up, biology students? Mr. Holloway here. In today's video, we're going to talk about how to build these evolutionary trees that you keep seeing in my videos, using both morphological and genetic evidence. Before we were able to read and analyze the universal genetic code of life, we had to rely exclusively on morphology to determine evolutionary relationships. Morphology refers to the physical form, shape, and structure of an organism, and could also include things like developmental patterns. When using morphology to build an evolutionary tree, we focus on what are called derived characters, traits that define a group because they evolved in the common ancestor of that group and were passed to all descendants of that common ancestor. The four-legged body plan of tetrapods is a great example. Having four legs defines all these animals and isn't something you find in any other group but this one. So if you find an animal with four legs, you know for sure that it's a tetrapod. The amniotic egg is a derived character that defines this group. Having hair is a derived character that defines this group. Specialized meat-eating teeth is a derived character that defines this group, and retractable claws is a derived character that defines this last and smallest group. That means that our cats here inherited all these derived characters, as we can see in this table, whereas our kangaroo only inherited 3, and the frog only inherited 1. If we had another animal, like a horse, that had all these traits, but not these two, we would know that it belongs somewhere in the clade Mammalia, but not in the clade Carnivora. Now let's build our own tree. Here in this table, I've got a list of derived characters, and the X's indicate which organisms possess these traits. Take a second to pause and make some predictions about how you think our tree is going to look based on this data. According to this data table, all these organisms have a dorsal nerve cord, or what we might call a spinal cord, and a notochord, which is a flexible rod that runs parallel to the nerve cord. All but the lamprey have a jawbone and bony vertebrae. These bony vertebrae develop from the notochord we just talked about in all these organisms, but in lamprey, it remains just a notochord. All but the lamprey and the tuna are also tetrapods with a four-legged body plan. The kangaroo, monkey, human, and snapping turtle all have an amniotic egg, in which embryos develop inside a protective membrane called an amniotic sac. Kangaroos, monkeys, and humans also have hair or fur and mammary glands, the glands that produce milk to feed offspring. Only the human and the monkey have a placenta, which is a special organ that helps a developing fetus to feed, exchange gases, and excrete waste. Humans also have shortened canine teeth, and the hole where our spinal cord comes out of our skull, the foramen magnum, is far forward, right underneath our skull, rather than toward the back. In most tetrapods, this hole is in the back of the skull, because most tetrapods walk on four legs, and have a spine that runs roughly parallel to the ground, so it makes sense that they would evolve to have a spinal cord that comes straight out the back of their skulls. Even monkeys have a foramen magnum that is farther towards the back of the skull, because while they can stand on two legs, they don't regularly walk around that way. Humans are the only group of organisms in this table to possess this derived trait. Based on the data I have, humans possess more of these derived characters than any other taxa. The taxon that is most similar to humans in terms of these particular traits is the rhesus monkey, so we can connect them like so. Labeling the tree like this indicates that this trait, having a placenta, evolved in the common ancestor of monkeys and humans, and that common ancestor is represented by this node, or intersection, here. That makes this clade the placental mammals. Next most similar to the two taxa we already have are the kangaroos, so we will connect them like this. This node now represents the common ancestor of humans, monkeys, and kangaroos, basically the common ancestor of all mammals. Once again, I will label our tree with the traits that define the branches I have so far, then repeat this process again for our next most similar taxon. That would be the snapping turtle. Connecting like so. Now this is the common ancestor of all the taxa with an amniotic egg, making this clade the amniotes. Next is the frog, which I will connect like this. Here is our new common ancestor at this node, and it was in this ancestor that the four-legged body plan first evolved, which defines this clade as the tetrapods. Next is the tuna, and this common ancestor is the first vertebrate, and also the first fish with a jawbone. This clade we formed represents the vertebrates. And last is our lamprey. No jaw, no spine, kinda gross, and way down here at the bottom, this represents the common ancestor of all chordates, all organisms with a dorsal nerve cord, or spinal cord to you and me. That common ancestor, by the way, was likely something very similar to this famous little creature here, Pikaia. Pikaia evolved during the Cambrian explosion some 530 million years ago, which we discovered due to this fossil here, which was found in the Burgess Shale Formation, which I talked about in one of my previous videos. Evolutionary trees like this tell a story, relating the steps involved in the evolutionary process that eventually led to organisms like us. This particular tree tells the story of chordates, who must have first evolved in the ocean, given that both these earliest diverging lineages belong to other ocean-dwelling creatures. We can see that a bony spine evolved first, then bony limbs, 4 of them to be precise. But these creatures still had to live near water because they hadn't yet evolved an amniotic egg. That would evolve next, allowing some tetrapods to move away from the shores and farther onto land. Hair evolved next, then a placenta, which allowed pregnant mothers to carry their developing young internally for longer periods of time before giving birth. And finally, organisms like us evolve, who walk upright and whose big brains allow us to invent things like tools and eventually computers for watching videos like this one. That was a pretty straightforward tree we just built. There wasn't really any uncertainty as to how it should be built, largely because of the specific traits I chose for that example. But real life is usually a lot more messy and ambiguous. Take our 3 domains of life, for example. It's only 3 branches, so you would think it would be pretty easy to make. But taking a look at this chart here, you might notice it's not as straightforward as the last tree. These 3 traits here indicate similarities between bacteria and archaea, suggesting that we should build our tree like this. In this case, bacteria and archaea are sister groups, and eukaryotes are the outgroup, which kinda makes sense since bacteria and archaea are all single-celled prokaryotes. But these 5 traits here indicate similarities between archaea and eukarya, and 5 similarities is more than the 3 similarities we identified previously. Moreover, these 5 similarities are arguably bigger because they have to do with how these cells read, replicate, and act on the universal genetic code contained in their DNA, and that suggests that we should build our tree this way. In this form, our tree tells us that archaea and eukarya are the sister groups, sharing a more recent common ancestor than either group shares with bacteria. And this is actually the scientifically accepted way to build this particular tree. Overall, there are more and bigger similarities between eukarya and archaea, even if there are still a lot of important similarities between archaea and bacteria that we can't ignore. But that's just life, I guess. We can also use genetic similarities to help us determine evolutionary relatedness. In this diagram here, we see an evolutionary tree based on a set of genes called Hox genes. These are some of the genes that control body development in animals. They help our cells figure out which end is the head, which end is the tail, where eyes should grow, all that kind of stuff. Without getting into too much detail here, each colored box represents one of the Hox genes. Even without knowing anything about these genes or what they do, or even what the heck kind of animals these are right here, we should be able to recognize similarities. These 2 groups obviously have very similar sets of genes, and the sheer number of copies of each sets them apart from the rest. Clearly very closely related based on those shared genetics. This one here doesn't have as many copies, but it has all the right colors, so it also must be pretty closely related to those other 2. These 2 taxa are nearly identical in their Hox genes, meaning closely related again, and these two down here are kinda close as well. Just like we did with derived characters, we can look for similarities in genes and genetic sequences to help us start identifying key similarities that influence how we build our trees. Genes code for protein molecules, and these proteins are made from sequences of building blocks called amino acids. By comparing the order of amino acids in the proteins built by different organisms, we can also kind of compare the genes that code for those amino acid sequences as well. Hemoglobin is a protein made by all vertebrate animals. In case you're curious, this protein's job is to carry oxygen in the bloodstream. This is an example I've mentioned before, but if you compare the hemoglobin proteins from different vertebrate animals, you can figure out who's related to who, because closely related groups will have very similar hemoglobin. That's because less time will have passed since those two groups shared a common ancestor, and that means less time for mutations to creep in and change these genes. More time means more mutations, more differences in genes and the proteins they code for. Looking at the animals listed here, we can see that the genetic evidence supports what we already thought to be the case just by looking at the morphology of these animals. Humans and gorillas are most closely related, with the fewest differences in this amino acid sequence. Then the monkeys, with a few more differences, then the mouse with even more. Those are all our mammals. Next, the chicken, then the frog, who has the most differences and therefore shares the most distant common ancestor with humans out of all of the animals listed here. So which is better for determining evolutionary relationships, morphology or genetics? Well, the scientific community commonly accepts that genes are more reliable than morphology for several reasons, not the least of which is the fact that convergent evolution has a way of producing organisms that look similar, and which may even behave similarly and occupy similar niches in their environment, but which are quite distantly related at the genetic level. Convergent evolution is when distantly related lineages evolve into similar forms because of similar environmental and selective pressures. Here in this diagram, we see a bunch of examples of the convergent evolution between marsupials and placental mammals. Even though the animals in each row look similar, all these animals are more closely related to other animals in their own column than they are to the animals in their own row. This mole is more closely related to this wolf than it is to the marsupial mole right over here. And that's because marsupial mammals and placental mammals are two totally separate groups of mammals, about as different as you can possibly be and still be mammals. Another interesting example of convergent evolution is the case of sharks, dolphins, and ichthyosaurs. All three live in the ocean and occupy similar niches, and have similar morphology as a result of similar selective pressures. But they belong to very different evolutionary groups. The shark is a fish and has been evolving along with fish for millions of years. The ichthyosaur was an ocean-dwelling reptile who lived at the time of the dinosaurs, one that evolved from ancient reptiles living on land. It is most definitely not a fish, even if it looks like one. And if it was alive today, genetic analysis would reveal this difference even more strongly than the fossils left behind do. And finally, dolphins and porpoises are mammals, not fish, and not reptiles. Those are all pretty obvious examples. I mean, honestly, it's pretty tough to mistake a fish for a reptile or for a mammal. But when we start getting into the real nitty-gritty of evolutionary relationships, genetics can help us to clear up a lot of ambiguity that morphology alone cannot. For example, the tree on the left, tree A, is based on morphological evidence, and the tree on the right, tree B, contains the exact same mammals, but arranged based on genetic evidence. Individual groups have been color-coded in both trees to make it easy to see the differences between these trees. As you can see, some relationships remain the same, but there are several places where groups got moved around a lot after the genetic evidence became available, and the color coding helps us to see this reshuffling. And we tend to think that this version, the one based on genetics, is probably more accurate. Ultimately, scientists use both morphology and genetics to help us sort out these kinds of puzzles, and the trees produced are one way to tell the story of how the diversity of life evolved on Earth over time. Hopefully at this point, you feel like you understand something about how these trees are made, and could even try your hand at making one yourself. And with that, I will bring our video to a close. Thanks for watching, everyone.